Hello, my dear students, and welcome back. With your exams just around the corner, I know things can get a bit stressful. So today, we're going to tackle a huge chapter, protein and cheminformatics. But please don't worry, we're going to make this super simple and quick. Ready? Let's dive right in. Just imagine this, Beta. Our bodies are like a giant library with lakhs and lakhs of books, and each book is a protein. Now, if you needed to find just one specific book to understand a disease, how would you even start? Especially with no search bar. It sounds impossible, right? Well, that's the exact problem scientists face with so much biological data. But what if we had some computer superpowers? In this quick explainer, we're going to see exactly how computers help us understand proteins and find new medicines. It's actually a fascinating story, and it's going to be fun, I promise. All right, let's get into part 1, decoding all the secrets of proteins with the help of computers. This whole field is called protein informatics. Now, don't get scared by the big name. Trust me, it's not as complicated as it sounds. For your exams, just remember this simple idea. Protein informatics is basically using computer tools to find and understand all the secret information that's hidden deep inside a protein. You can think of it like being a detective, but for molecules. So what's in our digital toolkit for this detective work? Well, the first and most important tool is something called ProtParam. Just think of it like a cool app on your phone that instantly gives you the complete biodata of any protein you're studying. It tells us some really important things. Let's look at them one by one. First up, we have the Isoelectric Point, or PI. Now, this is just a magic pH level where the protein is perfectly balanced. It has no overall charge, positive or negative. It's at its most stable, and you could say its happiest state. This is super useful for scientists who are trying to separate and purify it in the lab. Next is the Aliphatic Index. This sounds complicated, but it just measures how much space is taken up by a few specific amino acids. The only thing you need to remember for your exam is this. A high index score means the protein is very stable. Even when things get hot, it can really handle the heat. Okay, Becho, pay close attention to this one. This is a very common exam question. The Instability Index. It does exactly what its name says. It predicts if a protein will be stable in a test tube. And the rule is so easy to remember. If the score is less than 40, our protein is stable. Good. If it's more than 40, it's probably unstable. So less than 40 is good, more than 40 is bad. That's it. And last for this section, we have the gravy value. What a funny name, right? It actually stands for Grand Average Hydrophobicity. It's a big name for a simple job. It tells us if a protein is a water lover or a water hater. And all you need to know is that a low gravy score means the protein is hydrophilic. It loves to be around and interact with water. Okay, so now we have the protein's biodata. We know its properties, but that's only half the story. The real secret to how a protein works is in its 3D shape. But predicting this shape is really tricky. So scientists use a kind of flowchart, a map, to figure out the best way to do it. Now have a look at this map. The computer is like a detective here. It starts by asking a simple question. Do we already have the 3D structure of a protein that's very similar to this one? If the answer is yes, great, we're in luck. We can use that known structure as a template, a process called homology modeling. But if the answer is no, well, then we have to use more complex methods to build the structure from scratch. This flowchart shows you the exact logic the computer follows. So, we now know how computers help us find a protein's 3D shape. That shape is our target. It's like a lock that's causing a disease. So how do we design the perfect key to open that lock? Well, that brings us beautifully to part 2, cheminformatics. This is where chemistry meets computer science to find our new medicines. And again, here is a very simple definition for your exam. Cheminformatics is just using our computer superpowers to solve chemistry problems. And its biggest, most important job is helping us discover and design new drugs. So why do we even need computers for this? Well, just look at this number. There are over 219 million. That's more than 21 times crueler different chemical substances known to us. Can you imagine searching through all of them by hand to find the one right medicine? It would take thousands of years, but our computers can screen them in seconds. You know, making a new medicine is a very long and very expensive journey. It can take 10 to 15 years and crores of rupees just to bring one single drug from the lab to the pharmacy. Cheminformatics is a real game changer
because it makes this process so much faster and cheaper. It helps us find the best possible candidates on the computer first, before we even go into the lab.
Now for another star topic for your exam. This is a must-know. It's called Lipinski's rules. I like to think of it as the final exam that any potential medicine has to pass. It's a set of simple rules that helps us filter out all the molecules that probably won't make good oral drugs. And what's the magic number you need to remember? It's 5. So easy, right? That's why it's also called Lipinski's Rule of 5. See? You won't forget that, Bacho. So here are the rules. A good drug candidate that you can take as a tablet should have no more than 5 hydrogen bond donors, no more than 10 acceptors, a molecular weight under 500, and a log P value of less than 5. Now this log P just tells us how well it dissolves in fats compared to water. But listen carefully, this is the most crucial part. A molecule is allowed to break one of these rules. But if it breaks more than one, it fails the exam, it's out. Okay, quiz time. Let's do a quick check. A molecule has a weight of 600 Daltons. Does it pass this part of Lipinski's exam? Think about it. The rule says the weight should be under 500, so 600 is one violation. This molecule has failed at least one test. It could still pass if it doesn't break any other rule, but it already has one strike against it. See how that works? It's quite logical. You are all doing wonderfully. We are almost at the finish line. Let's just quickly wrap up with 2 final must-know key terms for your exam. Ready? First question, what is a pharmacophore? You'll see this term come up a lot. So a pharmacophore is not a real molecule you can hold in your hand. It's an idea. It's an abstract 3D map. It's like a blueprint that shows all the essential features a drug must have and where those features must be located to fit perfectly into its protein target, just like a key fits perfectly into a lock. And the final question for today, my dears, what is a lead compound? A lead compound is a chemical that has passed all those initial tests. It's showing promise. It has the right kind of activity we're looking for. It's like a finalist in a big competition that gets chosen for further development to become a real hero drug one day. And that's it. We did it. We've covered all the key concepts of this entire chapter in just a few minutes. You are all absolutely brilliant. Just remember to study smart, not only hard. I have full faith in every single one of you. You are going to ace your exams. All the very best.